Okay, so here we are. This is the second piece of the renal system lecture. So far, we've talked about how you create filtrate, so filtration, glomerular filtration, and the factors that affect glomerular filtration. Now what we need to do is talk about how you take that filtrate and you turn it into urine. Okay, so remember the general process of this thing is force everything through, force the liquid and everything through, everything except for proteins and large substances like that. And now we get to be picky about what comes back in. And that's how you maintain homeostasis, by regulating what comes back into the body. Okay, so the two processes we're going to talk about are tubular reabsorption. Remember, that means taking it back from the tube into the blood. If it's in the blood, you get to keep it. If it's in the tube, it goes away. You excrete that one. Okay, so reabsorption, bringing it back in, a secretion, putting it into the tube so you can excrete it out of the body. Okay, so let's talk first about how you reclaim water and salt. Okay, so here is the glomerular capsule. The very first part of the tube is called the proximal convoluted tubule. And the proximal convoluted tubule is the location where most of the water and most of the solutes are reclaimed back into the body. Okay, so most of it's going to come back within that very first initial segment. You then have what's called the loop of Henle. So here is the loop of Henle. You have a descending limb and an ascending limb. That is also very important for reclaiming salt and water. Okay, so we'll go through that, and then it'll go into the distal convoluted tubule, which will then dump into the collecting duct. The collecting duct is the location where the ADH, which is antidiuretic hormone, is going to act on. So let's look first at reabsorption in the proximal convoluted tubule. So remember that at this point, we've just forced everything through, and now we're going to pull things back in. The proximal convoluted tubule is very highly modified to make it very good at reabsorbing things back into the body. Most of the filtrate that was pushed out at the glomerulus is going to come back in. So especially most of the water you really want to hold on to, the most of that water, unless the blood volume is very high or you're very, because you're very well hydrated or something like that. Generally, most of the water is going to really come back into the body, and because that way you get to keep it in your cells. Okay, so let's look a little bit. I know this is anatomy kind of, but I do want to sort of touch on why the proximal convoluted tubule is so very, very good at its job. The first is the type, The first is the type of epithelial lining it has. It has simple cuboid epithelium. Remember, when you have when you have epithelial tissues that are simple, one single layer, that makes them very easy to reclaim. And then on top of that, they also have microvilli, and microvilli are little hair-like extensions. And so the more microvilli you have, the more surface area you have, and the more surface area you have, the better or more efficient the absorption will be. Okay. So let's look at how you reclaim water. So if you think about it, you have a liquid, which is plasma. You take that plasma and you force it through the holes. Most of the solutes are going to go along with the water. That means that the concentration of solutes in the plasma is now going to be the same as the concentration of the solutes in the filtrate, because the filtrate was created from the plasma. So they're going to have the same concentration. That means that they're isoosmotic to each other. They are isotonic. So there won't be any net movement of water between them. There's no osmotic pressure for the water to leave the tube. So how do we fix this? How do we make it so that we can get the water into the body? And the answer is we're going to actively pump salt. Okay, so please note that we are not actively pumping water itself. What we are doing is using salt pumps, and anytime I say the word pump, that means primary active transport. We're going to use primary active transport to pump a bunch of sodium out of the tube and into the extracellular fluid so that it can be reabsorbed back into the blood. And if you're pumping a bunch of salt, and by the way, the chloride has an attraction to it, so it's going to go along too. If you're pumping a bunch of salt outside of the tube, now what you've created is a concentration gradient. And if you increase the concentration of the salt, that means that relatively speaking, that'll be a lower concentration of the water, and solutes suck. So water will flow out of the tube following the concentration of the higher salt outside of the tube. So when you're talking about reclaiming water, what it does is it uses active pumps to create a concentration gradient that will then draw the water out through a passive process of osmosis. Okay, so the water is going to be drawn out because it's saltier outside of the tube than inside of the tube. And that is the same as for the loop of Henle as well. Okay, so we're not actively pumping water anywhere. Okay, so most of the water is going to be reabsorbed in the proximal convoluted tubule. But now we have the loop of Henle, and the loop of Henle is set up to maximize the amount of water that is going to be drawn back into the body. Right, so let me first go over the structure of the loop of Henle, and that's going to lead into a discussion of the countercurrent multiplier system. Okay, so here we have this proximal convoluted tubule, this structure right here, that is the loop of Henle, and it's a very sharp turn at the bottom. And so it has, and if you think about it, here's the flow of the filtrate here. So here is your descending limb, and then here is your ascending limb. So we have a descending limb and an ascending limb, and they're right next to each other. Now this ascending limb has a region which is called the thick segment, which is not permeable to water. That's important. Okay, so you have one limb that is permeable to water, water can get through it, and one limb that is not permeable to water. Water cannot get through the ascending limb. Okay, so when you have them so that they're right next to each other, that means that the environment is, they both share the same environment on the outside of the tube. Okay, and then that's going to be surrounded by peritubular capillaries to help draw anything that comes out back into the body. Okay, so let's go through the gist of the countercurrent multiplier system. So first of all, let's take this scary name and break it down. Okay, so counter. What does counter mean? So for example, if I say I have a counter argument, I have an opposing argument, an opposite argument, and then a current like flow, like think about like a current in the ocean or a current downstream. That just means a movement of liquids. And then multiplier refers to exponential effects, okay? So what we're doing is we're creating currents that go opposite to each other. We have a descending limb and an ascending limb, and the fluid in them is flowing opposite, counter to each other. Ah, brilliant. Okay, so when we're talking about the countercurrents multiplier system, I like to start with the ascending limb first, because to me that makes most sense in my brain. So we're going to start way over here first, but remember that the fluid is flowing down, going around the loop, and then coming up. Okay, so starting over here in the ascending limb has a thick segment which is not permeable to water, but what it does have is a bunch of salt pumps. Okay, so it's got a bunch of sodium chloride pumps, and it's going to be actively, when I say pump, I mean primary active transport. Okay, so it's going to be actively pumping out salt. That's going to make the environment outside of the tube very, very salty. But in this region here, because it is not permeable to water, water cannot get through, no water is going to enter back into the tube in this region. Okay, so here we have an external environment that is very, very salty outside of the tube. Now going back over to the descending limb, as the liquid flows through, and then this is still you know, this is still fairly liquidy at this point. As the liquid flows through, because it is more salty outside of the tube than inside of the tube, it has a higher concentration of solutes outside of the tube, and solutes suck, the water is going to then flow out of the tube via osmosis, and as the water leaves the tube, what is left? The solutes, right? So as the water is flowing out of the tube, that leaves solutes behind, and as it goes back up through the ascending limb, those solutes, the salt, will then be pumped out. Okay, so now what we have is a positive feedback loop. The more salt that is secreted by the ascending limb, the saltier it is outside, the more water will be drawn out, the more salt is available for the pumps to pump out, the more salt that comes out, the more, you know, more. Okay, so now what we've done is we've been able to maximize the amount of water that is pulled out of the renal tube so that it is able to go into the blood, the peritubular capillaries and the Vasa recta that surround it, in order to keep and hold and keep that blood for the body. Okay, so don't lose sight of the fact that the function, the purpose of the countercurrent multiplier system is to increase urine concentration, which just means there's a lot of solutes in there because there's not much liquid, and reabsorb maximum amount of water back into the body. Okay, so if we have a positive feedback loop, that's a snowballing effect, more and more and more and more. So when is the absolute max? Okay, when does the maximum amount of water we can reabsorb using the system reached? Okay, any ideas? Okay, so at what point have we reached the maximum amount of water that we can reabsorb? It has to do with the fact that these salts, they are pumped by carrier proteins, and carrier proteins exhibit saturation, right? So at some point, okay, it's usually about 1200 milliosmoles. What is it? 1200, milliosmoles. There we go. That's 1200 milliosmoles. At some point, we reach a point where those salt pumps are pumping as fast as they can, but it doesn't matter if there's more salt available to it, it can't pump anymore. And so that's going to put a cap on how much salt is in the external environment, which would then put a cap on how much water is drawn out. Okay, so you have that positive feedback loop until you reach that maximum where the salt pumps are saturated. I like how all of those concepts just come back. Okay, so now let's talk a little bit about the capillary beds that surround this. So there are two different types of capillary beds. You have the peritubular capillaries, which surround these areas here, and for the loop of Henle's that go way down into the juxtamedullary nephrons, they have a different blood vessel, which is called a Vasa recta. Okay, and you don't need to know the term. I'm not going to ask anatomy questions. But I would like you to understand that the function of this Vasa recta is to help reinforce the countercurrent multiplier system. And it does it because as the water is drawn out, okay, so if you're looking at this picture, here's the blood flow. Okay, so remember the blood flow is the same, it kind of follows the loop of Henle along here. So you've got the descending area and an ascending area here. So it'll kind of flow in the same general shape as the loop of Henle. Okay, so as the blood is flowing along, if you look at the black arrows indicate salt reabsorption. Okay, so diffusion of salt back into the blood. The salt will be reabsorbed, and as you see the numbers here, see how they increase? They were increasing because the concentration of the salt is increasing as the sodium is flowing from higher concentration to lower concentration. So you're putting the sodium back into the blood. But as you go into the ascending limb right here, what happens is Because the sodium is higher here, the lower here, it's going to cause the sodium to flow right back out. So it went in, but then it came right back out. And because you still have more concentration, higher concentration of the solute here, you're still going to be reabsorbing the water even then. So the gist of this, I know that was a little confusing, but the gist of it is what you've created as a salt trap, so that you're reclaiming water back into the body, but you're keeping that nice salty environment so that the countercurrent multiplier system can continue to work. That's the main gist here. Okay, so let's see. Yeah, let's talk about ADH. So ADH, again, ADH stands for Antidiuretic Hormone. And remember, a diuretic makes you pee. So antidiuretic stops you from peeing. So what it does is it helps increase the concentration of urine, and does so by taking the water out of the urine. So you'll have lower volume. And so what happens when you are dehydrated is that your body is going to secrete ADH, and it'll secrete the ADH, and what it does is it acts on the collecting duct. Okay, so at this point, you've already done reclaiming water through the proximal convoluted tubule, and you've already reclaimed water through the countercurrent multiplier system. If you need additional water, you're very dehydrated, now you're going to claim extra water under the influence of ADH at the collecting duct. Okay, so let's talk about how that works. So ADH is going to be, okay, so remember this is sort of an endocrine review here. ADH is going to be produced by the hypothalamus, and then it'll be stored and then secreted from the posterior pituitary. Okay, so if you are dehydrated, that'll cause the hypothalamus to produce the ADH, which will then cause the posterior pituitary to increase the secretion of ADH. Those ADH then go to travel all over the body, but their target organ will be the kidneys. And in those kidneys, they're going to increase the amount of water, and that's what's going to make it so that your urine is more concentrated. And so when you're really dehydrated and your urine is really, really yellow, it's really yellow like that because you're still getting rid of all the waste products, the urea, but you've reclaimed most of that water, so you have very small volume, and that small volume will be very concentrated. That's what the ADH does. So let's talk about the specific mode of action. So ADH, what it does is it doesn't actively pump or anything like that, the water. What it does is it makes the collecting duct more permeable to water, okay, able to pass through. And the way that it does that is by causing little vesicles that are stored inside of the cells lining the collecting duct to fuse with the plasma membrane of the cells. And now what we've done is we've inserted more of those aquaporins, which is just a fancy way to say water channels. So we inserted more water channels into the cell membrane of those cells lining the collecting duct. That's going to make it easier for the water to flow through, but what actually pulls it out is, remember that the water is still following the solutes. It's being drawn out by the salty environment surrounding the collecting duct. Okay, so all this does is make it easier for the water to go through. It doesn't actually draw it out. What draws it out is the salt that's already present surrounding the collecting duct. Okay, so it'll insert those water channels and it'll make it more permeable for the water to get through. Okay, so I'm going to go ahead and stop it for now, and we'll come back. We'll talk about glucose reabsorption, tubular secretion, and then the role of aldosterone.